Very well. So most everyone here, very good. You typed in good morning. If you typed it in before I started this recording, please type it in now. Now we take roll because it won't record it until after, I've initiated it. So please post good morning again, even if you've already done so. Thank you, thank you, thank you. All right. On the screen, I'm sharing the office hours. So make a note. And again, this will be in the recording now that we have it. And you'll see that I've tried to vary the time for the office hour. Mondays, 10:30 to 11:30 a.m. So today, as soon as I can set up the Zoom address, I will post it. I will have these office hours also posted in Canvas, uh, in course information. I will have them posted in course information so that you can access the office hours for this fall. If there's any change or if I have to cancel or what have you, I will notify you through Canvas. I will post an announcement with regard to that so that you can see that. Today, we're starting with the tissues of the body, and it looks something like this. So if you have your outline or access to it on your device, we begin here today, and we're going to go through epithelial tissues. We're going to go through connective tissues and some characteristics thereof, continuing on down into bone, blood, nervous tissue, and muscular tissue with epithelial glands and tissues, etc. If I can begin, I'll touch on the integument today, but I'm not sure I'll quite get to that entirely. Now, last week, I had ended with meiosis, and for your testing purposes, meiosis, you have to know that it is reduction division and utilizes the creation, it's utilized in the creation of a sperm and an egg. We've got two major phases to meiosis, meiosis 1 and 2. And each meiosis 1 and 2 also have their prophase, late prophase, metaphase, anaphase, telophase, prophase, metaphase, anaphase, telophase for meiosis 2. Um, meiosis 1 uh, for women occurs prior to birth, forming the egg. Meiosis 2 is finished off as the woman begins to ovulate, as she begins to ripen each egg, then meiosis 2 is completed. Meiosis 1 and meiosis 2 for men are both completed in the production of sperm. And we will talk in depth more of this during development. But for now, that's what you need to know with regard to meiosis. So with the tissues of the body, remember that there are four primary tissue classes: epithelial, connective tissue, nervous, and muscular. In microscopic examination, keep in mind you are trying to interpret a two-dimensional image into a three-dimensional construct of the tissue. So when you examine the diagrams that you're studying for the practicum, look at them and imagine this two-dimensional representation that you see as an image actually has 3 dimensions to it in actual tissue. Since we are not doing microscopy in the lab, this is not going to be so much an issue for you with regard to microscopy, because you're not going to have to focus on an image during the exam, but everybody will have the same view of these images. And again, all of the images can be found in the textbook, in the atlas, and in your lab manual. And I suggest you start going over those images and commit them to memory, because on the practical exams, you will be asked to do so. And let me see what we've got going here. Just checking the chat. Lots of people saying good morning still. Thank you. If you haven't said good morning yet, please do so, as this is how I take the role for the course when you come in. All right. So I'll stop this share and have a new one there. All right, on the screen, you should see chapter 4, part A, tissue, the living fabric. And we begin by discussing microscopy of human tissue. Remember that under the microscope, tissue is going to be fixed with a solvent. Fixation is mummification. You're mummifying a tissue because this tissue will be preserved in a paraffin block. And these paraffin blocks will be sectioned and cut into very thin slices to transmit light or electrons, depending on the process that's it's undergoing. And then they will be stained to enhance contrast. Now, the 2 types of stain that we use in this are hematoxylin and eosin, and I will type those in the chat box. Hematoxylin and eosin. These are the 2 primary stains that are used in histology. Hematoxylin is a purplish blue. Eosin is an orangey pink. We call them an H and E stain. So on the slides, if you ever see them in the lab, and it says H and E stain, that's what it means. And so we stain all tissues in order to be able to see the detail that they present on examination. And this is an entire process. And in fact, you can make a career of being a histotechnologist. It's a really good gig, I think. I might have been interested in pursuing that myself had I not gone on into clinical medicine. And the job involves coming in very early in the morning into the laboratory and taking these tissues that have been preserved overnight in a run in a machine. So the process of fixation is completely automated now, but it still requires that you be a histotech to section, actually, produce the cassettes with the paraffin blocks that you section and make the slides and stain them so that the pathologists can examine them as soon as they come onto their shift at 7:30, 8 a.m. in the morning and begin reading the slides. And usually you're done with your job by noon. So you come into work and you start working at, say, at 4, and you go through the whole run and everything, and you're out by noon. So it's not a bad job, and it's quiet. There's not anybody really looking over your neck. You have to be self-directing in this job. Very meticulous, and you can sit there and listen to your music while it's going on, and it pays very well. At the time that I was in the hospital, I think it had begun paying like $65,000 a year, and I'm sure it's probably gone up since then. So you might want to look into that. There is a program, I believe, still at Mount San Antonio College, Mount SAC, that still has a histology program locally. So that's where you can get certified and trained and then maybe go out and find a position somewhere. All right. So when we compare electron micrographs and transmission, we have here a light micrograph showing the H&E stain with the bluish purple to orangey pink. Electron micrographs give us more 3 dimensions. So that's the big difference between light transmission and electrons. Light transmission microscopy is more 2-dimensional. It's exclusively 2-dimensional. Electron, 3-dimensional. So here we're looking at a comparison of cilia, it looks like here. Yeah. And you can see more detail with the electron micrographs. You can also train to become an electron microscopist. That's a highly specialized field. And I do know that at Cal State Fullerton, at least there, you could go through certification training to do so. When I was a grad student at Cal State Fullerton, I went through that training. And so I learned how to use an electron microscope. Really interesting. And again, it's another field that pays really well. At Pennsylvania Hospital, where I was at, they were hiring at the time that I decided to move back to California. And there they had a position in electron microscopy. And that began at like 80,000. And that was a good 30 years ago. So I'm sure it's gone up since as far as the base pay for that. So that's another good career. All right, epithelial tissue is a sheet of cells that covers body surfaces or cavities. And there's 2 main forms, covering and lining epithelia. Covering external and internal surfaces. An example would be the skin. Skin is a form of epithelial tissue. And then we have glandular epithelium, and this is secretory. It's producing some product, and you find this in glands like salivary glands and sebaceous glands and sweat glands. Main functions of epithelial tissue are protection, absorption, filtration, excretion, secretion, and sensory reception. And we'll go into more detail as we get into the context here of the lecture. Now, I had already discussed desmosomes, hemidesmosomes, tight junctions, and gap junctions. Desmosomes in particular with pemphigus as to how the tissues line up and keep themselves arranged. Epithelial tissues are derived from 3 primary germ layers. And we'll talk more about those germ layers later on in development. On the screen, the characteristics of epithelial tissue is that they have polarity. This means that they have like an external surface or an apical surface, and then they have a deeper bottom surface. And you'll understand better when I show you a diagram on here coming up. And again, if you have any questions, just type them please into the chat box as we go through. The apical surface is the upper free side, and the basal surface is the lower attached side. It's attached to a basal membrane, a basement membrane. And the basement membrane is connective tissue. The basal lamina is the adhesive sheet, which holds the basal surface of epithelial cells to the underlying cells, to the basement membrane. And of course, both surfaces are going to differ in structure and function, and we will show you that. Up here, we see a photomicrograph showing epithelium. This is simple columnar because of its shape. If I enlarge the image a bit, you can see that we have an apical surface, and then we have the basal surface here. The dark oblate features are the nuclei located near the basal surface of a columnar cell. Columnar cells are longer than they are wide. Sometimes they have a modification on the apical surface like cilia or microvilli. Cilia, again, are going to wave things over the surface. I discussed this last week. And microvilli will be present to extend the cell membrane to increase absorptive surface features, to increase absorptive surface area. And again, we see microvilli in great richness in the tissue of the GI. The forms, as I've listed here on the lecture outline, are simple, stratified, pseudostratified, and transitional, and I will discuss and you will see here shortly the definitions of what it means. And in which content, one characteristic of epithelial tissue is that it is avascular. There are no blood vessels in epithelial tissue. You find the vascularization in underlying connective tissues. The epithelial tissue is therefore nourished and hydrated by osmosis and diffusion from the underlying connective tissues. However, epithelial cells are supplied by nerve fibers. So there are free nerve endings that come up into that tissue, and we'll show you some layers in a bit. Regeneration. Epithelial cells have a high regenerative capacity. That means that their mitotic index is high. They replace themselves like the lining of your GI tract replaces itself every 7 days. The surface of your skin replaces itself. Your hair continuously grows out and replaces itself. Red bone marrow, while not epithelial tissue, also has a high mitotic index and is producing blood. Now, simple epithelia is a single layer, that's it, of cells. Stratified are 2 or more cells thick. And so in stratification, it means just that. You get layers, strata, building up one on the other. Pseudostratified, let's see, do we have that coming up? All right, so here's an example of simple up on the screen versus stratified tissue. And so you can see the layers here. The most mitotically active cells are going to be along the basal surface. And as they divide and increase their numbers, they push upwards and outwards towards the apical surface. And so this apical surface is constantly being shedded and replaced by new cells from the bottom. Now, the process of aging is that our mitotic index slows down. So when you hear of somebody passing away and they've lived to be a great age, you know, they're 60 or 70, their mitotic index has slowed down and they're not replacing cells as readily as they did when they were children or in their teens or 20s. And so this is part of the normal aging process, is that mitosis slows down. So if we had a reliably extended mitotic life, our life expectancy would also extend. And so this is where that theory of human being capable and having the capacity genetically to live 1,000 years, because that seems to be the series of and the lifespan of how these tissues work. Now, again, some of you might be sitting there going, oh no, how can that be? How can they find something like that out? Okay, well, in the study of cancer, cancers are uncontrolled mitotic growth of cells, both epithelial and connective tissue cell lines. They just kind of come out of the bag in mitosis, and it becomes uncontrolled, and it spreads through the body and starts to create problems because of how it spreads and just continues to grow like weeds in a garden. They've studied cancer cells, and they have found certain cancer cell lines that have become immortal. Immortal. They seem not to have any cessation point, and the ones that are very close to being immortal seem, just to keep going on. For instance, there is a cancer cell line that they have kept since, gosh, it's like the late 1940s, early 1950s, from a woman, and I'm trying to remember the specific name. It's like LACMA or something like that, the acronym standing for the name of the woman from whom this cancer was harvested. Well, at that time, when they were studying it and they could grow the cells, it didn't take much effort to provide an enriched environment for the cells to grow in, they found that they have just kept replicating without end. Now, most cell lines have a finite shelf life as far as replication, not these cells. To this day, from the original cell line, there are still cells, and there's still research going on with this tissue. And so in this way, they have found out that yes, indeed, there is the capacity for immortality in our genes. Think about that. All right, the different shapes are here. Squamous cells are flat. So think of squamous as like squashed. Cuboidal, well, it's shaped like a cube, and the nucleus is round and set in the middle. In squamous cells, the nucleus is elliptical and sits in the center. And so on appearance, a sheet of squamous cells under the microscope sort of looks like a bunch of fried eggs all pushed together in its appearance. Columnar are longer than they are wide, and the nucleus, again, is oblate or ovoid and located near the basal portion of the cell. And let's see what's going here. Somebody typed into the chat box. Thank you very much. With regard to what I just talked about, the immortal cells, the HeLa cell line. Thank you very much. All right. The woman's name was Henrietta Lacks, and you guys can look that up and read all about it. Now, there's a crime associated with this because the woman's family has never gotten a penny in remuneration for the use of this mother's cell line from her cancer for themselves. Yet, research facilities, pharmaceutical companies have made billions of dollars, billions off of this. on their research and development and making other drugs, but nothing has been paid to the family. Nowadays, you can be careful a little bit with your genetic material, but you still kind of sign it away whenever they remove things in surgery. Uh, and of course, when people do the genetic testing online and they send in a sample of their skin, to find their genes, you've just given up your DNA and it's going to go wherever and however, if someone gets a hold of it. So just saying. And again, when you have questions, please type in the chat box. I'll be going back and forth with this. So simple squamous epithelium, they're flattened, cytoplasm is sparse. We see them in kidneys and lungs, and they form two tissues, endothelium, which is lining of the blood vessels and lymphatic vessels and of the heart, and mesothelium, a very specialized serous membrane found in the ventral body cavity, found in the thoracic cavity. We see mesothelium, particularly in the pleura, in the pleural membranes surrounding the lungs. And when this cell line goes malignant, meaning it becomes cancerous, the name is mesothelioma. Mesothelioma. All right, I keep hearing a bunch of noise in the background. Would you all make sure your mics are turned off, please? Kind of difficult. Everybody, please put themselves on mute. Thank you. All right. So up on the screen, here's an example of the simple. We see this in the lungs making up the alveoli. And the alveoli are small balloons within the lungs that form the structures through which gas exchange occurs between these air sacs and the overlying capillaries. And the gas exchange occurs by diffusion. And of course, we absorb we absorb oxygen and exhale and release carbon dioxide. Well, all of that has to pass across the squamous cell layer. More about the lungs later. All right, let's see. Now, in addition, on my outline, I put in with stratified that it can be keratinized or non-keratinized. What does that mean? Well, keratin is a protective protein. Keratin is found in stratified squamous epithelium, which makes up our skin, and it protects you. So wherever you make contact, wherever you touch your skin, it's keratinized. This is a protection. It helps to keep our water inside. It helps to keep out pathogens like bacteria, viruses, and fungi from growing within us. Certain other tissues that normally don't become keratinized can become keratinized, and that would be like the lining of your mouth on down through the esophagus. These are generally non-keratinized epithelium. So for now, keratinization occurs in the skin, and we see it non-keratinized in the mucous membranes of epithelial tissue. Cuboidal is going to be either single or stratified. There are rare examples in the body of stratified cuboidal epithelium, but for the most part, single layers, simple is the construct. And these are going to form the walls of very small ducts or glands, and of course, the tubules of the kidney. Here we see projected a diagram that shows simple cuboidal as it's set up with a photomicrograph, a light microscope photograph of the cuboidal tissues stained again with H&E, and you can see the coloration. Note the multitude of nuclei that you see here. And here's an example of where they're found within the organs of the kidney. So remember, as you study, you have to be able to recognize the tissue on a picture and be prepared to answer questions like identify the cell type of this tissue. And what do I mean by the cell type? Well, is it squamous? Is it cuboidal? Or is it columnar? That's almost always a question on the practicum. Identify the cell type. Then it'll be asking you, identify the tissue. And so looking at this, you'd say, oh, I know that is simple cuboidal. And so you write that down in the practicum. So that's the difference between those two types of questions on a practical exam. Also, I ask this in written form when you take your lecture exams and quizzes. Now, simple columnar epithelium, single layer of tall, closely packed cells. Some cells have microvilli and some have cilia. Again, microvilli are structures that increase the absorptive surface area of the cell. It's extensions of the membrane, and it's used to bring things into the cell. And we see a rich layer of microvilli in the epithelial lining the GI tract. And we will show you in more detail when we get to the GI later this semester. Some cells have cilia, and the cilia waves and moves things over the surface. Notably, coming up is my favorite tissue, and that tissue is ciliated. And the purpose of that tissue, well, it's to move things out from the deep lung, out into your throat, out into your nose to get rid of debris that you breathe in. Because we don't breathe in pure air. The air you breathe in is laden with ash from smoke particles, from petrochemical combustion, dirt, all sorts of things. And so we want to keep the lungs sweet and clean. Some layers contain mucus secreting goblet cells. Goblet cells produce mucus. Mucus is used as an adhesive and as a lubricant by the body. An adhesive in our respiratory tract and reproductive tracts because it traps pathogens. And so by the action of the cilia, it's able to wave globs of mucus that have entrapped this material up and out of our body. It's also used for lubrication along with serous fluid. So here's a note. Ciliated cells move the mucus. Found in the digestive tract, gallbladder, ducts of some glands, bronchi, and the uterine tubes. That would be the reproductive tract there, if you didn't know. So, diagram, it shows here, columnar epithelium packed up well, nuclei present. Again, note, nuclei are in the basal portion of the cell. The photomicrograph, again, with an H and E stain, shows us columnar epithelium with goblet cells. So these areas here that my pointer on the screen is being moved over, these are goblet cells, and they're producing mucus. Cells, goblet cells, do not have microvilli. Goblet cells do not have cilia because it's constantly releasing mucus. And so there's no point in having specialized features or adaptations on the apical surface because it's being replaced constantly. So each cell at its apical portion will not have that. Now it looks like it is here, but remember this is a two-dimensional form, and instead of being cut perfectly along the cell line, it could be cut at an angle. And so we see some areas represented more than others. Let me see, where are we at? Okay. Ah, my favorite. This again is my personal favorite tissue, pseudostratified, ciliated columnar epithelium. Many cells are ciliated, but you always include that in the name. So please make a note if you're writing this down that it's not just pseudostratified columnar epithelium, but it is pseudostratified ciliated columnar epithelium. We want to get the whole name in there. This tissue is moving material up from the lungs, mostly. So here we have a diagram. It's called pseudo-stratified because that word pseudo means false, falsely stratified. Now you look here and you think, oh, it looks pretty stratified to me. I mean, look at those nuclei. But let's look carefully at the diagram. Note that each cell is going to make contact with this basement membrane. Here's our basement membrane along where my cursor is moving. Each cell does indeed, and they've done many studies, electron microscopy especially, to show that each cell is going to actually touch the basement membrane. Now again, you see it like make a point here and make a point here. Well, that's continuing on behind these other two. So this cell here, while it looks like it stops there, it does not. It does make contact. Now, the nuclei can be just about everywhere, and that kind of gives us the illusion that there are strata. And so after they did studies, they said, well, it's not really stratified at all. It's actually a simple layer. So we'll just call it pseudo-stratified because it's falsely stratified in its appearance that we see here. On the photomicrograph, H&E stain again, we see this. We see the presence of goblet cells producing their mucus. And here we see cilia. Look how lush and beautiful that is. That's just gorgeous. You will be asked to identify specialized features on the practicum, such as cilia. So this is a very nice picture. And when we look under the microscope, some of the best slides look just like this. So if there's a pointer on the picture and it says, identify this feature, you would say, oh, that's cilia. If it points here and says, identify this cell or feature, you would say, oh, that's a mucus cell. That's a goblet cell. And if we say, identify the tissue, then you write the whole name down. Pseudostratified ciliated columnar epithelium. And if we said, identify the cell type, you would say, oh, it's columnar. So that's a lot of information there, but that's kind of how you need to organize your learning. Again, note cards are the way to go, I think. And it's very helpful. Again, you might be able just to sit there and memorize it uh, just by looking at it with all that in your head. Good for you. All right. Stratified epithelial tissues involve two or more layers of cells. They all regenerate. Stratified squamous epithelium, again, found in the skin. And it helps with high wear and tear, which occurs on the skin. An example of localized keratinization on the skin are calluses. Calluses. So if you work out, if you work construction, anything to do with your hands, you're going to form calluses on your hands. Well, that's a buildup of keratin. On your feet, if you run or you go walking a lot, you're going to develop a callus. Again, this is a growth of keratinized cells, and it's the way your body has of protecting you from wearing out these areas of high wear and tear. Guys and girls that play the guitar or any other stringed instrument, if the strings are steel, they'll notice that the tips of their fingers, after playing, become toughened and will form a callus. Any type of work you do. Um, Unusual, but I had calluses here on the lateral surface of my fingers after working in a pathology lab by just removing the plastic lids of formalin. And it was constant for me having to open up containers of formalin because these had the specimens from surgery, these had the biopsies from outside clinics. And so part of my job was helping to process this tissue for the fixation to come. But over time, and even after wearing a glove or double gloves most of the time, I still had a ridge of callus even today. And I haven't worked in the hospital like in over 25 years. So it'll stick with you. So here we have stratified squamous epithelium, an example of the skin. This one is non-keratinized, and looking at the photomicrograph, you see the nuclei all the way up near the surface. Here we can see that this is not keratinized because a keratinized example, there would be no nucleus. The tissue replaces the nucleus with keratin. So by the time we get to the apical layers, there's no nucleus. But here we can see it. So that's how we know it is non-keratinized. And this, of course, is lining the mouth. We also see it in the esophagus and the vagina, and of course on the cervix. Again, if you have any questions, put them in the chat box and I'll be checking. All right. Stratified cuboidal epithelium is quite rare. So most of it, like I said, in the body is simple. Stratified columnar epithelium, this is very rare, very limited distribution in the body, small amounts found in the pharynx, the urethra of the male, and some glandular ducts. Looking for now is transitional epithelium. Let's see. Ah, here we go. I skipped over it. Okay. All right. So transitional epithelium, we see this in the urinary bladder, ureters, and the urethra. The ureters are the structures that carry urine from the kidneys down to the urinary bladder. The urethra is the lining of the duct that carries urine from the urinary bladder outside of the body. So we see it in the urethra of both men and women. The urinary bladder, this epithelium, is within. Now, the basal layers of the cells are either cuboidal or columnar in shape. Most of the time it looks like cuboidal, but this tissue has the capacity of stretch. Here we see a relaxed form of transitional epithelium. You'll notice that the cells are like from columnar to cuboidal in shape. We see nuclei up near the apex, and of course in the midst, down below. In the lining of the urinary bladder, as the urinary bladder fills, this tissue has the capacity to stretch. And so it's called transitional epithelium because it transitions between these cuboidal columnar shapes down to a squamous shape when it's fully stretched out and it flattens. All right. And let me just check the chat box really quick. Nothing here? Okay. All right. Moving along, let's see here. I will come to the endocrine and exocrine and all of that in a moment. But right now, I want to seg into connective tissue really quick and come back to this. So I will stop this share. I'll pick up another share here. Let's see. Where am I at? All right, integument, connective tissue. All right, projected up on the screen, we have the other PowerPoint now that looks at connective tissue. Connective tissue is the most abundant and widely distributed of the tissues. Four main classes: connective tissue proper, cartilage, bone, and blood. Major functions are going to be binding and support. These are support tissues. They protect, insulate, they store, reserve fuel. And so, what tissue do you think in your body is storing fuel? Anybody? Put it in the chat box. What connective tissue stores fuel? When people get too much of it, they like to lose some of it. And someone's put in the chat box, fat. You are correct. But we don't say fat. We say adipose. Adipose. And the name of the cells are adipocytes, and we will get to that. Good. All right. The chart up on the screen shows us the comparison between all the tissues we're going to discuss here: areolar, adipose, reticular, dense regular, dense irregular, and elastic tissues. The cell lines are also present: fibroblasts, fibrocytes, adipocytes. Cartilages, three types: hyaline, elastic, and fibro, are made of chondroblasts on down to chondrocytes. Chondroblasts are in the growing stages and are found in growing cartilage as it develops. That suffix, blast, indicate an actively growing immature cell. Cyte indicates a mature end stage cell. Make a note of that. Other examples are bone, blood. Yes, blood is considered connective tissue. Cell lines of bone are osteoblasts, Keep in mind what I just told you about still growing and osteocytes, the end stage mature line, red blood cells in the blood, and then we have white blood cells. Red blood cells are colored red because of the presence of hemoglobin. Hemoglobin is a protein that carries oxygen and releases it in tissues that need it and will pick up carbon dioxide to carry away to the lungs. White blood cells have no hemoglobin, and this is why they're called white, because hemoglobin is a protein that is notably red, and that's because of the presence of iron. We'll get into that detail when we get to blood as a lecture, but that's it for now. All right, the matrix is the place where the tissues live. The matrix is what we call most of the connective tissue. For you see, in epithelial cells, in epithelial tissue, there's a great density of cells when you look at it through the microscope. In connective tissue, while you do see cells, there's not as many of them, and they're surrounded by stuff. It's surrounded by what looks like it could be a gel on inspection, and you're thinking about it, gee, what's this kind of empty space that we see here? But it's called a matrix, and the matrix is made up of all different sorts of things. It's mostly a protein sugar mesh in which it helps to support the cells. And indeed, the matrix supports them, so the tissue itself can bear weight, withstand tension, and endure a lot of orthopedic abuse, especially. So we see that in a lot of the joints. The ground substance is part of this matrix, and there's going to be fibers and cells. Fibers like collagen or elastic. And then, of course, the cells themselves. Ground substance is an unstructured gel-like material filling the space between the cells. And indeed, it's the cells that are producing and maintaining this ground substance. It's going to be largely fluid, water, interstitial fluid, with a glue for attachment, glycoproteins, with proteoglycans. These are the sugared proteins. And these are going to be adhesive as well, composed of large polysaccharides. That word polysaccharides means complex chains of sugar. Poly prefix mean many, saccharide means sugar. Examples of this are going to be chondroitin sulfate and hyaluronic acid. These are examples of the fibers I just mentioned, collagen and elastic fibers. Now, going to my outline where it says characteristics of fibrous connective tissue, I have listed the cells, fibroblasts, macrophages, leukocytes, plasma cells, mast cells, and adipocytes. Now, fibroblasts make collagen. Write that down somewhere in your notes. Very important factoid. Fibroblasts make collagen. Fibroblasts are also mitotically active. Fibroblasts are also mitotically active. So collagen, we see it a lot in connective tissue in certain types. Some don't have collagen. Skin, collagen. Skin is supported by connective tissue. So the actual skin itself is keratinized, stratified, squamous epithelium. But the connective tissue base is going to be made of connective, but the base is connective tissue. And so this connective tissue is going to either be like, let's see, it's full of collagen, it's full of fibroblasts that help support the skin. Fibroblasts, when we're young and as we age, are mitotically active. When we get to older age, this mitotic index drops. And so there's less collagen made for replacement. And that's when you begin to see the wrinkles. All right, let's just discuss the blast cells and site cells for you. All right, let's see. Now, moving on down, macrophages are phagocytic cells that eat dead cells, and we see these in the blood, and they're also within like areolar connective tissue that help to keep it sweet and clean. Leukocytes, granular and agranular, are the white blood cells, and these are part of the immune system, and it's your tissue's response to injury. Leukocytes, leukos means white. Plasma cells, and I don't see plasma cells up on the screen, but plasma cells are a specialized white blood cell that are going to make antibodies. And we will talk about that when we get to the immune lecture. Mast cells initiate a local inflammatory response against foreign microorganisms they detect. Mast cells are rich in the substance histamine. Histamine. We'll type that in the box. Histamine. Now, there's a class of drugs that fights against histamine. Anybody? Type in the chat box. What is that class of drugs? What have you heard as being antihistamine in nature when you see drug advertisements or you've heard of drug advertisements? Good. You've even given me the specialist drug name, Benadryl, allergy medicine. Excellent. Allergies. Histamine has a huge role in the allergic reaction of our body. Antihistamines counteract their release. So mast cells are rich in histamine. Sometimes, though, they can all come out of a bag at once. And this is when we suffer the reaction of an allergy. We are having an immune response to something coming in. And so in order to get rid of it and jump on it right away, the body releases histamine. And this is what causes the allergic reaction. Good. We'll talk more about this a little later, but you must know that little factoid now. Adipocytes are the fat cells. Adipocytes store nutrients, and there are two types of adipocytes, white fat and brown fat. White fat, actually it looks yellow like chicken fat, like you see on a chicken breast or chicken thighs when you remove it. That's kind of the color and consistency of the adipose in your body, the white fat. Brown fat is rich in mitochondria. Brown fat we see exclusively in the newborn and infants. So up through about a year of age, the baby has brown fat. This is truly what baby fat is. And being rich in mitochondria, it's producing a lot of ATP for energy to help keep the baby warm. And so that's why it's important so to have the baby born full term so that it has this richness of brown fat to help keep it warm because it's such a small thing, a small person. Babies that are not full term don't have this rich layer, and so they're in danger of hypothermia, of just getting over chilled because they don't have that capacity, that energy to sustain warmth for themselves. Again, brown fat has mitochondria, and we see that again in babies, the newborn up about to a year of age or so. And then someone asks, mast cells work with plasma cells in the inflammatory response, right? Yes, it does. Very good. We'll talk more about that when we get to the immune system. White fat is actually yellow. It looks yellow. You know, I've seen many, many bodies for autopsy, and I can indeed tell you it is yellow, but we call it white fat because it is not rich in mitochondria. All right, let's see. Moving right along. All right. The fibers I've listed, collagenous or collagen, reticular, and elastic, and we'll come back to those in examples that I'm going to show you. Ground substance or matrix, glycosaminoglycans, acronym is GAGs, proteoglycans, and adhesive glycoproteins. I've mentioned those three already. Now, next page, at the top of the page, it says types of fibrous connective tissue. Well, we have loose connective tissue, and again, CT stand for connective tissue. So learn that because I will use that in exams saying this type of CT, that type of CT. So you got to know CT means connective tissue when you're reading it. So there's loose, there's dense, and in the dense, there's regular and irregular, areolar, reticular, adipose, and they're the example of dense regular and dense irregular. Now, areolar